Yes.
Ethan says he on the field trip and then he emailed you his question and his assignment.
Okay, thank you. Any field trips to those guys go on? Don't have a stapler. If you just fold it over, we'll get it that way. Yeah, just do that. I'll get it and I'll staple it for you. What the hell am I doing? Okay, I guess everybody that's going to be here is going to be here. We got questions up front. We got assignments over on the side. We'll take it from there. So would help if things were visible. So review session after class, if anybody's interested. With the exam coming up a week from today, does anybody want to have any sort of review session next week?
The questions that we turned in from 2 weeks ago.
I'll go over them right now. I'm can imagine on some level the questions are the review sessions, or could be.
When you can hand back
As soon as I can. Life has been a little crazy. My plan is to try to get everything caught up this weekend, maybe even tomorrow. And then I can leave it out on the bulletin board in Marshall here, or up at the dairy, if you want it before Monday.
up the dairy spring.
Okay. I can, as soon as I get everything in there, or I'll put it on the board out here so you have it before Tuesday. Everybody get an assignment 10? So I know you two just turned in assignment 9, but I wanted to show you where the end of the exam should have been. So you know where that's supposed to be. The corrected one is also up on Blackboard. So after question 6 is the coverage for the next exam. Anybody interested in extra credit? Monday, we're going to try to get rid of all the hay and pigeon shit and all that stuff from the top half of the old livestock barn. So I'm going to try to do things from 4 to 5. We should be able to get 2 spreader loads out, and that should be the bulk of it. I don't know if it'll be all of it. And our next exam is a week from today. So, question for the class. We have an opportunity. There's a heifer that's pretty banged up and doesn't seem to be getting any better. There's thoughts of getting rid of her, selling her, but given how rough she looks, we're probably not going to get anything for her. So Justin suggested her as our dissection. So she's limping around okay. So there's no real hurry per se. But we could, in theory, do it next Tuesday. If you want to do it before the exam, or would you like to do it after the exam?
We'll do all our stuff during it, but help us during the exam.
You'll look at some of the bones and the tendons, and the later you get, you're going to have, you could look at the heart. We got the heart. There's no lungs on this exam. What else we got? We got bones, muscles. If you want to look at some of those, the different veins and arteries.
Before the exam.
If we did it next Tuesday, those are some of the things on the exam that might be related. Should we try to arrange it for Tuesday?
Yes.
Okay. So we'll meet up at the dairy. I'll confirm that on Blackboard. I got to talk to Justin to confirm that that's what we'll do. I talked to him this morning. He goes, yeah, anytime you want, we can do that. So we might try to save a leg or two for future classes. And then I guess things like the kidneys, the lungs. We did the digestive tract and nutrition. What else we got left? Metabolism, you really can't see that. The brain, nervous system will be later lectures as we move forward. So we'll see if we can make that arrangement, plan on that, pencil that in. I will confirm that on Blackboard when I talk to Justin. Other questions or concerns? Okay, questions from the last class. Different questions we got last time. What are the other ways to regulate cardiac output. So what are the two principal ways to regulate cardiac output? Cardiac output is a function of what two things? Or what is cardiac output? Let's start with that. Amount of blood per unit time. So when we're thinking about cardiac output, we need to have the number of beats per minute and then the volume of blood per beat. You multiply those two things together and then you get the volume per minute. So most of the time, cardiac output is measured in milliliters or liters per minute. Other ways that we might regulate cardiac output, there might be some opportunity with peripheral resistance. So certainly with training will influence the two things, the beats per minute and the volume per beat. I'm not really sure what else we might do to influence the amount that's pumped out of the heart every minute. Why does it seem that jerseys are more prone to milk fever? Oddly enough, I'm working with some students. The new NRC came out, NRC Dairy Nutrition came out this past December. A couple of students that have an interest in advanced nutrition. We're reading a chapter a week and meeting on Tuesday or Thursdays to discuss it. The last chapter we read was about transition cow problems. Their suggestion that why the jerseys and other color breeds have more problem is the amount of calcium they put out in the milk. They put out 20% more on a daily basis than the Holsteins do. So whatever regulation or set point in that breed is putting out more calcium. So they are more likely to exhaust their blood pool, their exchangeable pool. They're going to exhaust it faster. So you're looking like 10, 15 extra grams per day coming out of those jerseys as opposed to the Holsteins. So that's why they're probably more prone to it. They're just having different genetic set points for calcium concentration in the milk. At least that's what current research is suggesting. How does a muscle relax? What's the big thing that has to happen for a muscle to relax? First thing that's got to happen is we got to put the calcium away. We put it back in the sarcoplasmic reticulum, put it back in the T tubules. We pull that out. The actin and the myosin can't cross-link anymore. They can't grab, and the muscle relaxes. So usually there's no more nervous signal that's pushing the calcium out into the sarcoplasm. The T tubules will scavenge that calcium up, put it away, and everything slides back to a more relaxed position. So you got to stop the nerve impulse. You've got to stop or put the calcium back in the T tubules. Can you go over muscles that work in opposition again? So a skeletal muscle can only pull in one direction. The other muscles tend to pull in multiple directions. Heart and smooth muscles can be branched, whereas our myofibrils are, in our skeletal muscles, they're linear. All they can do is contract. They can't push. So looking at the diagrams, looking at what the muscles do, these in my mind might be ones that work in opposition. So yeah, I meant to print this slide out for you. I forgot, sorry. Or I got distracted doing other things. So things that might work in opposition. So one pulls the head up, one pulls the head down. If we look at the longissimus muscle, that runs from the base of the head all the way to the tail. So in one part, it's going to move the head around. It's going to move the head up versus the sternocephalicus. It looks like it's going to pull the head down. And then you've got the abdominals on the back, on the stomach, ventral side working against the longissimus in the back. So depending on where you are, some of these, you're going to have multiple muscles working in opposition going through. Is this perfect or 100%? Maybe. So given the diagrams, those are the best answers I can give you. I think I gave those to you at the lab, but those are the same thing written out. So we'll make sure that gets posted so you can play with that. Is that any questions on that?
Well, are you going to ask us those on the test?
What was the,
Like,
what question was it for the homework? I forget how I phrased it. However I phrased it, I'm going to have to be exceedingly lenient. So whatever the phrasing is on the homework assignment, somewhere in that is the way I'll ask it. I think I said what pairs are in opposition. I might just ask what the hell is opposition? Why do they have to work in pairs?
So we're not going to need to know like the exact pairs or non-positions in the test?
I wouldn't worry too hard about that.
Okay.
So you need to know what each muscle does, and from there you should sort of be able to guess a few pairs. So maybe I'll give it as extra credit. But whatever I put it as the homework assignment question will be the way we'll address it. And I won't be that. I don't know that perfectly. I don't expect you to know that perfectly. What is Dr. Beatty's flow chart? A proposed early postpartum hypocalcemia cascade. Oh, this is our diagram here. So what does this diagram tell you? Where the calcium might be having its effect. So calcium in the muscles. What else does this diagram tell you? Big thing with our transition cows is one thing leads to another. If we get milk fever, and that's usually the first thing that happens, because it happens at that switch. Right at calving, we switch from feeding, supporting the fetus to supporting the calf. We switch from the uterus to the mammary gland, and we start pulling calcium out, and we lower that blood calcium, that exchangeable pool that leads to the hypocalcemia. One thing leads to another. So you can see that there's a train wreck potentially happening here. Hypocalcemia, DA, uterine retained placenta, metritis, ketosis, all are going to compromise that animal moving forward. So we need to prevent that hypocalcemia at the top. But then we reduce the likelihood of those other things happening. We'll spend more time on that in 220. How one thing leads to another and what we need to do to manage or prevent all these train wrecks from happening. So that's the cascade. The important thing is one thing leads to another. And we need to prevent hypocalcemia to reduce the likelihood of all those other things happening. And the worst part about it is the hypo or subclinical, where they're kind of a little lethargic, they're a little shaky, but man, we don't have to give them bottle. But they still have low blood calcium. that low blood calcium will affect, not completely, but will inhibit those two functions of smooth muscle and predispose that animal to problems. So we may not even see the hypocalcemia and there'll still be a problem. Can you go back through the main points of the blood path? So we're thinking about the red blood cell and its movement through the system. Where do you want to start? Right atrium. So where does it go through next? It's going to go right ventricle. What's it going to pass through to get there? AV valve. Right ventricle. Where do we go from there? Right side of the heart goes where? What is that pump to? Lungs. So to get to the lungs from the right ventricle, we first go through what valves? Semilunar valves. Then what vessel is the first vessel we go to? Pulmonary artery. It splits right and left. We go to the lung capillaries. We come back to the heart in what? Eventually the AV valve, but what's before that? Pulmonary veins, right and left, bring things back. Left atrium, AV valve. Left ventricle, semilunar valve to what vessel? Aorta, going out. That's going to branch off into 100 different directions. Go to the capillaries of the body and come back through two vessels called the what? What's the main return to the right atrium? Vena cava. So you've got the right side of the heart pumping to the lungs, artery, capillary, veins. You got sets of valves that you move through. And then the left side pumps aorta to the body, back to the two vena cavas, bringing blood back to the heart. So where is blood oxygenated? Where does it start to be oxygenated? In the lungs. So from the lungs, pulmonary vein, left side of the heart, aorta. You get to the body capillaries, what happens? Deoxygenated. So those capillaries, we change from oxygenated to deoxygenated. We come back to vena cava, right side of the heart. Pulmonary artery is all deoxygenated. So those are the parts I think are fair to ask you. So how would you like to put those in order? You want some sort of matching on the page, or you want me to lay something out on the table and you put the parts in order? Matching. We have done kind of hands-on thing before COVID started. I was like, you go, here's all the parts, put them in order, take a picture of it, and that's your answer to the question. And you call back up, somebody else comes up, plays with it.
Yeah.
So maybe we'll give you both options and give you the higher of the two grades. For a hands-on school, I can't test you all the time just on paper. So we have to get away from the multiple choice stuff. So those are the questions. I'll try to get those posted this afternoon. I've got meetings till about 3 o'clock. I might get things posted in that 3 to 4, unless the Dean had made a very odd request on Tuesday, and it's like, didn't I do this already? So I made this huge spreadsheet and it said, here. Now I have to explain it this afternoon, so should be fun. So we talked about answering the question just like we're going to do on the homework. The homework's a little shorter this week, given that you're studying. We're just going to focus on the lungs. So answering the question, how do we breathe? So what parts do we move through? That's your list there. So how does the body move? What are the parts? How does form follow function? Where does gas exchange occur? At the very end of the pathway in the elastic parts, the ducts and the alveolar ducts and the alveolar sacs. We'll show you how that works. We talked about the mechanics, what muscles are involved, how we increase and decrease the pressure in the lungs, and then the air moves from a high to a low, assuming we don't hold our breath in some way, shape, or form. We talked about how much air is in the lungs and how we describe those different amounts of air, the 4 volumes and the 4 capacities. And then how do you see these in a diagram? For a person normally breathing, not necessarily normally, we've got, this is how much, this line here, how much air is in the lungs, how much potentially we could put in the lungs. Any questions on any of that? Well, let's see if we can finish up the lungs today and get a good whack out of the kidneys. So how does a cow breathe? We've got this diagram here. This is a cross section of the alveoli. So what are all the features or functions that we see in this diagram that are going to help us breathe, help us stay alive? So the first thing we have around those alveoli is an elastic connective tissue. So those alveoli can stretch and get bigger, snap back, get smaller to push the air out. The diaphragm goes down, makes those things bigger, brings in the fresh air at the end there. So we have that connective tissue. It's elastic, it's also thin. So we're looking out, we got this big area of gas here. We don't have a lot of distance for that gas to diffuse. So what do we have here? The lining of the alveolus. You've got single cell layer is the outline or outside of the alveolus. They're flat, they're thin, so we can move through, oxygen can move through very quickly. If they were big and globular, it would take many more times to get through that. So we've got, just like the capillary bed, we've got a single layer of cells that the gases have to move through. They've got all the parts there, the nucleus, all those things to repair the cell. Big guess of what that is. What does this look like right here? Little barbell. What do we know that's shaped like a little barbell? Cross section of the red blood cell. The capillary is right here. We talked about that net of capillaries around the alveolus. Those capillaries are so small and so thin that the red blood cells are moving it through sideways. What's the advantage of that? All that hemoglobin is right up next to the alveolus. We have that one single layer there that makes the capillary, the cell going around it. Very little distance to go through the lung cell the connective tissue, and then the capillary cell. Very little distance for that oxygen and CO2 to travel. And then presenting the red blood cell that way, all that hemoglobin is as close to the air as it can be. You turn it this way, we've got this is close, circle, this is farther away. You turn it sideways, we get all that stuff right next to where the gas needs to go, where the gas needs to come from. What's this? A white blood cell. As you can imagine, there's a lot of things in the air that may not be healthy to us that we're breathing in. I'm trying to prevent the person I'm living with right now with COPD from getting COVID, because it'll probably kill her. I certainly don't want to have that on my head. But you've got those white blood cells. They are no longer in the blood. They are out in the body. If anything comes in, they are right there to attack it. In the commercials, you'll hear them called eosinophils. Does anybody remember why we call it an eosinophil? Because of the way it reacts with a certain dye. So I think they use the word eosinophil in the commercial because if people were killing their red blood cells, I think they'd get upset. But we call them eosinophils and we get around that understanding, and that ignorance allows people to be more comfortable. So form follows function. So we have a very thin layer that the gas needs to go through to get to the red blood cells. We've got this much. So diffusion is going to happen very quickly into the red blood cell, out of the blood. We have something that's elastic. That's going to increase in volume to bring in new air, collapse to push out the bad. Bring in the good, collapse the bad. The trachea, bronchioles are all rigid. That airspace is not going to change all that much. So the change in lung volume is down in the alveoli. We need that air to get there, and the best way to do that is to change the size, that elasticity of the alveoli. You've got those round surfaces covered in blood, so you're going to get the maximum exchange of gases between the air that comes in and the blood. So all these things in the white blood cells protect anything that might be inhaled. So form follows function, as it usually does in nature. So you've got all those parts working together to get things done. You can read through. All that stuff should be listed down at the bottom. Did somebody need that again? Top left where? This?
Yeah.
This is meant to be in the diagram showing what they're zooming in on. So you've got 3 alveoli here, and it's showing that intersection of the 3 right here. Does that make sense? So what is it? They do it with maps. Here's the area. We're going to put a square around it, and then we're going to enlarge it and show it to you. So we're putting a square around this intersection of 3 alveoli, 1, 2, 3, and showing you what the details are like for that section. So how does a cow breathe? Just like in other things we'll talk about, everything goes from a high to a low. You've got this diagram, it's already filled in for you. I pulled a bunch of stuff out to make it a little clearer. At least you should have this. So we've got the pulmonary alveolus. That's where the air comes in, that's where the air goes out. So oxygen is coming in, CO2 is leaving. In that previous diagram, we had that real thin layer between those. You're going to go from the pulmonary capillary to the pulmonary vein to the left side of the heart, back out to the capillary beds of the body. So that's all implied in this arrow here. We get to the tissue capillaries, where we drop off the oxygen, pick up the CO2, and then we head back to the heart, the lungs, where we make that exchange again. So everything moves from a high to a low. So if we go through and look at what the pressures are, or proportions are, of oxygen versus carbon dioxide. So compared to outside air, oxygen is going to be somewhere around 150, 200. Oxygen is going to be down like 1, 2. Well, because you have that CO2 leaving and being diluted by the oxygen, what's coming in or what's in the alveoli, 104 millimeters of mercury, partial pressure of oxygen, versus partial pressure of CO2 is 40. We compare that to the blood or the tissue. Tissue is going to be at 50 millimeters of mercury, partial pressure. So not a huge difference between the tissue, where respiration is occurring, where carbon dioxide is being generated, and what's in the lungs. Huge difference from what's outside. The air is less than 1% carbon dioxide. That's about 5, 6, 7% carbon dioxide in that location there. Depending on how hard we're working, we go from 104 down to 30 or less in the tissues. The harder those tissues are working, the lower that number gets. So the natural tendency is oxygen wants to flow in this direction, from the lungs to the tissues, and then carbon dioxide wants to flow high to a low from inside the body to outside. So that will happen if you let things sit for a while, but that's not nearly fast enough to keep us alive. So we have our lungs, we have our circulatory system to speed up that process. There was, what is it? They said at some point the oxygen atmosphere of the Earth was much higher, and that's what enabled the giant dragonflies, because they rely on diffusion. Insects, they rely on diffusion more, and because there was more oxygen in the atmosphere, they could grow bigger and still get done what needed to get done. Exchange that oxygen, get it into the body. So what we have coming back to the lungs, 40 for oxygen, 45. As it moves across the pulmonary capillary, everything exchanges. 45 is going to go to 40. 40 is going to go, or 104 is going to go to 100. So when we leave the lungs, oxygen is going to be around 100. PO2 is going to be somewhere around 40, which is sort of what we want as a low point in our blood. And we'll talk about that in a few minutes. So that's going to go through this heart, out into the body. No real change moving through those vessels. As it moves across the tissues, it's going to drop off oxygen, pick up carbon dioxide. So when you leave the tissues, you've got something, you dropped off 60 points, you're down to 40, which takes us back there. You picked up 5 going across. The smaller difference between carbon dioxide means it moves slower. The greater difference between oxygen in locations means it moves more aggressively, moves faster. So oxygen is falling down its concentration gradient to the tissues. Carbon dioxide is falling down its concentration gradient to the lungs. So everybody see those steps. So here's another expression of that. The alveoli, how does carbon dioxide leave the body? We know that oxygen is carried directly by the red blood cells. Carbon dioxide moves in a number of different ways. It can be dissolved in the water, just like a soda. There's certain reactions in the plasma, and there's also certain reactions with the red blood cell. So there's a number of different ways for carbon dioxide to be moved in the blood. It's not all about the hemoglobin. It is a little related, but not perfectly related. Those of you that take a more advanced physiology class will probably run into Duke's physiology. That seems to be the standard textbook that most schools use. And it's got probably 100-year history right now. Any questions on the lungs and how cows breathe? So what is a leukocyte? What does it do? White blood cell, what does it do? Defends the body. Depending on which type of white blood cell you are, you may have a different task in the army that defends the body. You may throw antibodies, you may throw oxidants out, you may engulf, you may destroy. Lots of different approaches. Where is the water in a cow? Where's most of the water? What space do you find most of the water? Intracellular. How much? 65% maybe of the total body water. Almost half of her body weight is water inside cells. So we want to defend that, and she will try to do that at all costs. When you mess that up, bad things happen. I can attest to that. Messing that up in wrestling, in high school, my electrolytes, my water balance, I spent 2 days puking. So don't want to do that again. So what are the electrolytes? What do we say electrolytes? What are we thinking of? What are the obvious ones? Sodium, potassium, chloride. Those are the minerals. Bicarb also falls into that category, but that comes and goes. That can be created and destroyed. Where do you find most of the sodium? Where do you find most of the sodium? Well, what's another, what we might define sodium in the body as the what? What? Primary salt. And that is what?
Like balances
It does balance things out. That's what it's there for. Most of the sodium is outside the cell. Sodium has a positive charge. So sodium is the primary extracellular cation with a positive charge. In that lecture I gave you on YouTube, you got all that sodium in the plasma, but you got very little sodium inside the cell because it's continually pumped out. Potassium is the opposite in some ways. Primary intracellular cation inside the cell. Where do you find most of the chloride? Extracellular. So you're thinking about the minuses. Chloride is the primary extracellular anion, negative charge. What's the primary negative charge inside the cell? What? Primary negative charge inside the cell that balances out potassium. Positive charge. Protein. So if we look at those charges, all the plus in an area in the body with all the minuses are going to add up to close to 300. 300 milliosmoles is what? Anybody remember? Isotonic. So those electrolytes, those protein molecules, the bicarb are all contributing to osmolality. They're also trying to control pH. The balance of the positive and negative charges are the control or determination of pH. How does the rumen best work? You've got all those reflexes and stuff that the cow controls, but she's also got to be oriented to gravity. A ruminant will not work in space without gravity. They need the gravity to pull the heavy stuff to the bottom of the rumen, to the reticulum, and let the air float up. If a cow was teleported to the International Space Station, she'd die of bloat very quickly because she can't get that gas out because it's not going where it needs to with gravity anymore.
So then why the caterpillar? If the gravity pulls.
Okay. So gravity usually works. There's a couple different types. One is the gas is produced so fast that they can't get rid of it fast enough. So production exceeds removal. So that's like if a cow got in and ate straight grain, that might blow her up. Another is you get what's called frothy bloat, where you've got like basically foam in the rumen. They can't eructate, they can't get rid of that foam because it's not developing into a big pocket. So what happens is that air pocket gets pushed down to the esophagus, goes sort of into the lungs and pushed out ahead of the exhale. You don't want foam going into the lungs, bad. So there's a reason that the gas is restricted in some way that's beyond just where it's located. So, or an animal goes over on her side, air bubble in the rumen is in the wrong place. Needs to be at the top of the rumen, not the side. It's on the side. She tries to eructate. All she's going to get is rumen fluid. And if that gets into her lungs, that's a bad day. So that air pocket needs to be in a specific location, and it's got to be free of froth and all that fun stuff. So let's move on to the kidneys. What do the kidneys do? So this was a picture that was presented to a friend of mine in med school on her first day discussing the kidneys. So what do the kidneys do? Make urine? Well, if we want to think about the job of the kidneys is to make urine. Does anybody know what this is? It's the Statue of David by Michelangelo. It's considered one of the greatest sculptures ever made. Looks reasonably lifelike. So we could say the analogy is that the kidneys are designed to make urine is the same as Michelangelo sculpting David to make marble chips. Urine is a byproduct of what the kidneys do. The kidney's job is to maintain the blood, which is equivalent to the statue. And then the waste products of that, the stuff that doesn't belong, the chips on the floor, is urine. So what do the kidneys do? They maintain the blood. They help with the gas balance. They help with all the nutrient balances. They make sure the pH is where it needs to be. Urine is the waste of this process. So it's a subtle difference, but I think it's one we should remember. The kidneys maintain the blood. They get rid of all the junk, all the excess that does not belong there. So sculpting is said to be, well, you just vision, in Michelangelo's case, David, and you take all the way the stuff that doesn't belong. Urine is the stuff that doesn't belong. So when we're looking at a kidney, and you should have these diagrams, one form or another, the blood is going to come in through the renal artery, renal meaning kidney, and the contents of the blood are going to leave one of two ways. Either it's going to be filtered, reclaimed, and come back in the renal vein, reclaimed by the body, maintained by the kidney, or it's going to be deemed waste product, and it's going to go out the ureter to the bladder and be voided out wherever the cow is standing. Has anybody seen like a recycling line for like construction waste or recycling stuff, where a whole bunch of stuff is dumped at one end of a conveyor, and a lot of people that may not have any other choices about their occupation sit there, and my job for recycling, I pull out all the milk jugs. Next person down pulls out all the cans. Next person pulls all the white glass. All the things that are important or useful get pulled out of that moving stream of stuff. And at the other end of that is the stuff that's junk, nothing that has no use to anybody picking on that line. Same way with construction debris. I pull out the wood, pull out whatever I can recycle, the metal, the sheet metal, any of that stuff gets pulled out on off that conveyor, and then the rest going off is really truly garbage. If you've ever seen this work on an industrial scale, it's pretty phenomenal. But it takes a huge human toll to make it happen. So we're separating the good, maintaining the blood, and then all the stuff that doesn't belong gets moved out to the bladder. So this is a kidney. This would be more representative of a kidney that you or I or a horse might have. It's not the same large configuration that a cow or another ruminant would have. And you'll see that in our dissection. We think of kidneys, you guys think of kidneys as sort of a kidney bean shape, right? That's not the way. The kidneys of a cow tend to look like a bunch of huge grapes. Still got that overall bean shape, but they got the big circular huge grapes that form the structure. So you're going to have the pelvis of the kidney, which is where the blood comes in, the blood goes out, the urine leaves. You're going to have two different layers. The medulla is more inside, and then you have the cortex, which is the area around the outside of the kidney. In this case, and what you're going to have in a cow is each of those little grapes is going to have its own cortex, its own medulla. So we already mentioned the artery, vein, and the ureter. One liquid in, two liquids out. So we're looking at our kidney. We're going to have something that looks a little bit more like that for our cows, where you have individual lobes associated with that. So we've got an ox there in the middle. We've got these individual lobes, and each of these grape-like, super grape size has a cortex on the outside and the medulla on the inside. Instead of one continuous like the horse, got the cortex running all the way around, we've broken it up into little sections. It still works the same. It still has the same basic unit, which is a nephron. So you're looking at, not showing the blood supply here, but we've got these individual sections that are going to filter the blood, manage the blood, and divide it into stuff that goes back into the blood. And then you have the duct collecting system that starts on the out here, works down to the medulla, and then drains into the duct system that finds that common ureter as you're going through. So we'll be able to see that in the dissection, how that lays out. And you can see as you dissect those pieces of kidney, the cortex versus the medulla. When you're looking at your kidneys in the body, you tend to have them staggered. So what's the advantage of staggering? One kidney's a little bit more cranial, one kidney's a little bit more caudal. What's the advantage of that? What if they were even? How would that diagram change? You'd have two arteries coming out in the same circumference. That's going to make that artery much weaker. By staggering them, you've got one opening in the artery here, one opening in the artery down here. So it has to do with structural integrity. If we put both openings opposite each other, the material that the arteries are made out of is not strong enough to handle that over time. So letting that there, we'll also see that there's the adrenal glands that sit cranial end of the kidneys. I'm not sure we'll be able to find those. We tend to lose those in the dissection. There can be a lot of blood, and we're leaning that animal on its back, trying to find it. It can be a little bit of a challenge, especially if there's some fat in the area. So zooming in on that, you can see that a little bit better there. How things are a little bit stronger. Again, with our cow, we have those lobules that are all going to work toward that common drain there. So why don't we stop there, take your break. We'll start at 2 after again. And we'll talk about the functional unit of the kidney, the nephron.
Are they putting an elevator in this building?
Not that I'm aware of. What do you think? What- Well, this building was designed to have a second floor. So there's enough steel in these ceilings to support another level, maybe two. They never got around to doing it. I don't know if you're probably not here in January. It gets cold enough, those iron girders start to contract. You get these big loud booms in here as they shift. So there is a staircase in the back there that you can go up. There's sort of a semi-attic in the back, but if they were to put in a second floor, which I doubt they're ever going to do, they'd have to put in an elevator. That's just requirements. Okay, so when we're thinking about the kidney, the functional unit we're dealing with is called the nephron. And this is the nephron. So when you hear people studying kidney function, they're nephrologists, nephrology. So this is the functional unit. This is what separates or maintains the blood, separates it into clean blood or good blood, and waste product. So all of the nephrons sit in this orientation. Doesn't matter what the shape of the kidney is. You've got these parts of the nephron are going to be up in the cortex. And then you've got these parts down here are going to be down in the medulla. So the blood comes in. We have a filtering process that occurs here. We reclaim all the stuff that we want. Whatever is designated waste flows out of the nephron. So if we've got one in and two out, the venous is continual circulation. We start with a dead end up here, and then we have a tube that dumps into the bladder. So that nephron, all of them are going to sit with the cortex parts and then the medulla parts. So your parts of your nephron, that's the orientation there, sort of looking at the nephron, how it all fits together. So what parts are we starting with? You have the blood supply coming in and it goes into a capillary bed. That capillary bed is a ball inside what's called, there's a glomerulus, that ball of capillaries, that's going to sit inside the Bowman's capsule, or the start of the nephron. In the glomerulus, the Bowman's capsule, the blood coming in, almost all of the plasma and its contents are removed from the blood. So what you have leaving the Bowman's capsule is blood that is almost all red blood cells, white blood cells, platelets, and then just enough liquid to allow it to move. Everything else is taken out. So if we're looking at plasma, what's taken out? Water, salts, proteins, maybe not so much proteins, but certainly amino acids, glucose, anything that's in there, hormones, is going to be moved into the nephron. We go from the Bowman's capsule to the proximal convoluted tubule. So the first one we come to, so that's the proximal. You'll notice on the other side we have a distal. From the proximal convoluted tubule, we work our way through the cortex. When we get to the medulla cortex junction, we transfer into the loop of Henle. That loop goes down and comes back up. Once we leave the medulla, we transition to the distal convoluted tubule. From there, a bunch of distal convoluted tubules gather together. You can see here's one, here's one, here's one, here's one, here's one. All gathered together and share a common collecting tubule that leads to the ureter. So a whole bunch of nephrons will service one collecting tubule. Everybody with me? So again, looking where that is in relationship to the cortex and medulla in a cow. Looking at that a little bit closer. Here's another representation of the same thing. The nephron, we begin with the Bowman's capsule, proximal convoluted. So you can see it's a little bit more convoluted than the previous diagram. We leave the cortex. We drop down into the loop of Henle. You can divide it into descending and ascending loops if you wanted to. We're going to just call it the loop of Henle. We're not doctors. Then we go into the distal convoluted tube. And then the collecting duct, collecting tubule on the other side. So all the nephrons are going to collect together in the ureter. So you've got nephrons all over the place doing what needs to be done. Now, to really get the full picture, with a nephron, you have surrounding it another secondary capillary bed. So the blood comes in through the artery, work our way down. You've got a capillary bed here where all the plasma, or most of the plasma, is removed. That's what will be cleaned. That's what will be maintained. Then we come out through a short, what do we call this? Afferent arteriole. And then we have another capillary bed that surrounds that nephron. The idea is we've pushed all the plasma out into the nephron. The nephron's job, just like I told you in those recycling places, is to pull out everything that is good or useful and put it back in the blood. So once it's filtered out, the nephron has to recognize it and say, hey, that's useful. Hey, that's good. The stuff that is waste, the stuff that is in excess, the stuff that's not supposed to be there, or the stuff that, well, I don't know what the hell that is, stays in that nephron, ends up in the collecting tubule, ends up in the urine. So the job of the nephron is to pull out the stuff it needs to reconstitute that blood. So when the blood leaves that surrounding nephron, goes back to the common blood supply, all the junk that's not supposed to be there, the marble chips on the floor, stays in the nephron, goes into collecting tubule, and Michelangelo's David leaves through the veins. Cleaned, managed, maintained. So that's the diagram you have. So do you have this diagram?
Yes.
Okay. So I used to draw this up, but when we went to COVID, I had to put together a diagram so we can work on this through here. So what I've done is I've laid out the nephron, just kind of pulled on it, right? So Bowman's capsule, glomerulus, proximal, Henle, distal, collecting tubule. I just laid it out flat. We're not going to get into the details like you would in med school, but the general flow of stuff that needs to happen has to move from here to here to be kept. If we want it, it's got to move from here to here. That nephron and the capillary bed around it. This is going to lead back to the heart. This is going to lead to the bladder and be voided. So the idea is we pull out the stuff that we need. So what leaves is essentially plasma. So we have plasma to start here, right at the Bowman's capsule. What we have leaving the glomerulus is sludge. All the red blood cells, the white blood cells, and just enough plasma to move it. So we've got sludge moving through that capillary bed. So whole blood entering has now been split. Effectively plasma and cellular components. And what happens at each point along the way, and we'll show you some of those diagrams, I don't think we're going to get to them today. Each different spot of the nephron is responsible for reclaiming certain components. We're taking them out of the nephron and putting them back in the blood. So what you have at the end of it all, you go from plasma to urine, and you go from sludge to reconstituted blood, cleaned blood, maintained blood at the other end. Is this making sense? So the nephron is going to focus on the solutes. Whatever's needed, the electrolytes, the glucose, the hormones, anything that needs to be in the blood is actively removed from the nephron and put into the blood. So it's all about moving solutes. And then what do we know about water? Water is going to follow passively. So we move the solutes, the water will follow that solute from the nephron back to the blood. So actively managing whatever solutes we want to save in the plasma, and then water will follow in an isotonic manner to the blood. So by the time we get moving stuff from here to here, moving it down, moving it down, moving it down, we should have reconstituted isoosmotic, proper pH blood at the other end. Does this visualization help make that point? It's not the way it's shaped, but I think it helps understand how things move through. So this is how it's shaped. So you can see our loop of Henle is surrounded by the capillaries. So the nephron is going to move stuff over back to the blood. Whatever is designated as not useful, whatever is designated as junk, keeps going down the nephron. So this leads to a point of messing with your friends. Does anybody here like to torture your best friends? So I have a group of gentlemen. We went to Newton High School. We are called the Newton Boys Reunion. We get together on a regular basis. We have one member, we'll call him Bill, who takes pride in pretty much eating anything. I can eat anything. He's not so crazy as the lab tech I worked with at Michigan State who used to eat light bulbs.
Light bulb?
Yeah, he says the key is you gotta chew them.
They're glass.
The glass, yeah. Dave Main was crazy. So we were getting ready to go on a camping trip. This is a long, long time ago. And Paul had an antibiotic he got, I'm not really sure how, but he goes, I want to feed this to Bill. And so me and him are like, well, how do we do that? And it came down to, we stuck it in a donut, and I bet Bill he couldn't fit the whole donut in his mouth. Bill, not to turn down a challenge, playing with his ego, he fit the entire donut with the pill in his mouth. Now this drug here, if you're taking rifampicin, it can cause certain body fluids, such as urine and tears, to become orange, red in color. A benign side effect that can be frightening if it's not expected or prepared for. Has anybody dealt with this drug before? So we went on a camping trip. We got him to eat this just before we went out. This was a January camping trip. We used to go camping all year round. So along the way, Bill slipped and fell, which is kind of an interesting side effect. And when we got to our campsite, as we often do, males will go find a convenient tree and write our name in the snow. Well, Bill started writing his name in the snow and started screaming. He thought he damaged his kidneys. Me and Paul are laughing our asses off because we didn't know what we had done. So after the fact, he's like, man, that was cool. You really got me good. So that's kind of the friendship we have. But if you're thinking about kidney function, what has happened with this rifampicin? Got filtered through the urine because what? Nephron didn't know what to do with it. That's not my job. I don't bring that stuff back into the blood. So as that antibiotic gets to the blood, it gets filtered out because the nephron doesn't recognize it as something useful. Same thing happens with penicillin. It gets filtered out from the kidney, which is why we need that daily dose of it. Early studies with penicillin would actually collect the urine, pull out the penicillin, and recycle it into other humans. Urine is probably actually cleaner than the broth it came from originally. So they have to, purify it, bring it back. But the nephron doesn't know what to do with this. And because it's bright red in color, you can mess with your friends. So none of the parts of the nephron recognize that as essential, and they let it go. So it ends up in the urine. It gets filtered out in a number of different locations. So we'll start there next time. Let's pencil in going to dissection next Tuesday. I'll double check the winter. I think it's supposed to be fairly warm. We don't want it too warm. The smell gets too bad. But we'll plan on that. I'll check with Justin to see if that works. Does anybody want to be part? What I try to do with this, since we only have a limited time, is we put down the animal and we get it set up. Does anybody want to be part of that, or you just like to have it done when you get there? Okay, if you want to, need to be there about 10 o'clock. So we usually put down the animal, try to get as much blood out as we can, and then we start the initial cuts to open it up. So plan on that for Tuesday. I will confirm that on Blackboard.